Cell structure. Units of measurement used in histology. The study of histology frequently involves the measurement of microscopic distances. The units used for this purpose are as follows. 1 micrometer or micron m equals 1/1000 of a millimeter, mm. 1 nanometer, nm, equals 1/1000 of a micrometer. Cells, tissues, and organs. The human body, like that of most other animals and plants, is made up of units called cells. Cells can differ greatly in their structure. However, most of them have certain features in common. These are described in this chapter. Aggregations of cells of a common type, or of common types, constitute tissues. Apart from the cells, many tissues have varying intercellular substances that may separate the cells from one another. Organs, example the heart, stomach, or liver, are made up of combinations of various kinds of tissue. Cell membrane. A cell is bounded by a plasma membrane, within which is enclosed a complex material called protoplasm. The protoplasm consists of a central, more dense, part called the nuc/eds, and an outer less dense part called the cytoplasm. The nucleus is separated from the cytoplasm by a nuclear membrane. The cytoplasm has a fluid base, matrix, which is referred to as the cytosol or hyaloplasm. The cytosol contains a number of organelles which have distinctive structure and functions. Many of them are in the form of membranes that enclose spaces. These spaces are collectively referred to as the vacuoplasm. From what has been said above, it is evident that membranes play an important part in the constitution of the cell. The various membranes within the cell have a common basic structure which we will consider before going on to study cell structure in detail. Basic membrane structure. When suitable preparations are examined by the average cell membrane is seen to be about nm thick. It consists of two densely stained layers separated by a lighter zone, thus creating a trilaminar appearance, Fig 1.1a. Cell membranes are made up predominantly of lipids. Proteins and carbohydrates are also present. Lipids in cell membranes. It is now known that the trilaminar structure of membranes is produced by the arrangement of lipid molecules, predominantly phospholipids, that constitute the basic framework of the membrane, Fig 1.1b. Each phospholipid molecule consists of an enlarged head in which the phosphate portion is located, and of two thin tails, Fig 1.2. The head end is also called the polar end while the tail end is the nonpolar end. The head end is soluble in water and is said to be hydrophilic. The tail end is insoluble and is said to be hydrophobic. When such molecules are suspended in an aqueous medium, they arrange themselves so that the hydrophilic ends are in contact with the medium, but the hydrophobic ends are not. They do so by forming a bilayer. The dark staining parts of the membrane, seen by M, are formed by the heads of the molecules, while the light staining intermediate zone is occupied by the tails, thus giving the membrane its trilaminar appearance. Because of the manner of its formation, the membrane is to be regarded as a fluid structure that can readily reform when its continuity is disturbed. For the same reasons, proteins present within the membrane, see below, can move freely within the membrane. Some details regarding the lipid content of cell membranes are as follows. As stated above, phospholipids are the main constituents of cell membranes. They are of various types including phosphatidylcholine, sphingomyelin, phosphatidylserine, and phosphatidylethanolamine. Cholesterol provides stability to the membrane. Glycolipids are present only over the outer surface of cell membranes. One glycolipid is galactocerebroside, which is an important constituent of myelin. Another category of glycolipids seen are ganglionosides. Proteins in cell membranes. In addition to molecules of lipids, the cell membrane contains several proteins. It was initially thought that the proteins formed a layer on each side of the phospholipid molecules, forming a protein phospholipid sandwich. However, it is now known that this is not so. The proteins are present in the form of irregularly rounded masses. Most of them are embedded within the thickness of the membrane and partly project on one of its surfaces, either outer or inner. However, some proteins occupy the entire thickness of the membrane and may project out of both its surfaces, Fig 1.3. These are called transmembrane proteins. The proteins of the membrane are of great significance as follows. A. They may form an essential part of the structure of the membrane, i.e., they may be structural proteins. B. Some proteins play a vital role in transport across the membrane and act as pumps. Ions get attached to the protein on one surface and move with the protein to the other surface. Some proteins are so shaped that they form passive channels through which substances can diffuse through the membrane. However, these channels can be closed by a change in the shape of the protein. A. Other proteins act as receptors for specific hormones or neurotransmitters. B. Some proteins act as enzymes. Carbohydrates of cell membranes. In addition to the phospholipids and proteins, carbohydrates are present at the surface of the membrane. They are attached either to the proteins, forming glycoproteins, or to the lipids, forming glycolipids, Fig 1.4. The carbohydrate layer is specially well developed on the external surface of the plasma membrane forming the cell boundary. This layer is referred to as the cell coat or glycocalyx. Membranes in cells are highly permeable to water, and to oxygen, but charged ions, Na, K*, do not pass through easily. The cell membrane. The membrane separating the cytoplasm of the cell from surrounding structures is called the CE/membrane or the plasma membrane. It has the basic structure described above. We have seen that the carbohydrate layer, or glycocalyx, is specially well formed on the external surface of this membrane. The glycocalyx is made up of the carbohydrate portions or glycoproteins and glycolipids present in the cell membrane. Some functions attributed to the glycocalyx are as follows. A. Special adhesion molecules present in the layer enable the cell to adhere to specific types of cells, or to specific extracellular molecules. B. The layer contains antigens. These include major histocompatibility antigens, MHC. In erythrocytes the glycocalyx contains blood group antigens. C. Most molecules in the glycocalyx are negatively charged, causing adjoining cells to repel one another. This force of repulsion maintains the 20 nanometers interval between cells. However, some molecules that are positively charged adhere to negatively charged molecules of adjoining cells, holding the cells together at these sites. The cell membrane is of great importance in regulating the activities as follows. A. The membrane maintains the shape of the cell. B. It controls the passage of all substances into or out of the cell. Some substances, consisting of small molecules, pass through the passive channels already described. This does not involve deformation of the membrane. Larger molecules enter the cell by the process of endocytosis described below. C. The cell membrane forms a sensory surface. This function is most developed in nerve and muscle cells. The plasma membranes of such cells are normally polarized, the external surface bears a positive charge and the internal surface bears a negative charge, the potential difference being as much as 100 MV. When suitably stimulated there is a selective passage of sodium and potassium ions across the membrane reversing the charge. This is called depolarization. It results in contraction in the case of muscle, or in generation of a nerve impulse in the case of neurons. D. The surface of the cell membrane bears receptors that may be specific for particular molecules, example hormones or enzymes. Stimulation of such receptors, example by the specific hormone, can produce profound effects on the activity of the cell. Receptors also play an important role in absorption of specific molecules into the cell as described below. Enzymes present within the membrane may be activated when they come in contact with specific molecules. Activation of the enzymes can influence metabolism within the cell as explained below. When a receptor on the cell surface is stimulated, this often activates some substances within the cell that are referred to as second messengers. Important second messengers are as follows. Adenylate cyclase, this enzyme changes the concentration of cyclic adenosine monophosphate, cyclic AMP, within the cell. In turn, this can lead to alterations in many functions of the cell, including protein synthesis and synthesis of DNA. Enzymes controlling cyclic GMP have effects that are usually opposite to those controlling cyclic AMP. Phosphoinositol, a phospholipid, affects calcium regulatory processes within the cell. Membrane proteins help to maintain the structural integrity of the cell by giving attachment to cytoskeletal filaments. They also help to provide adhesion between cells and extracellular materials. Cell membranes may show a high degree of specialization in some cells. For example, the membranes of rod and cone cells, present in the retina, bear proteins that are sensitive to light. Role of cell membrane in transport of material into or out of the cell. We have seen, above, that some molecules can enter cells by passing through passive channels in the cell membrane. Large molecules enter the cell by the process of enclocytosis, Fig 1.5. In this process, the molecule invaginates a part of the cell membrane, which first surrounds the molecule, and then separates, from the rest of the cell membrane, to form an encytocytic vesicles. This vesicle can move through the cytosol to other parts of the cell. The term pinocytosis is applied to a process similar to endocytosis when the vesicles, then called pinocytotic vesicles, formed are used for absorption of fluids, or other small molecules, into the cell. Some cells use the process of endocytosis to engulf foreign matter, example bacteria. The process is then referred to as phagocytosis. Molecules produced within the cytoplasm, example secretions, may be enclosed in membranes to form vesicles that approach the cell membrane and fuse with its internal surface. The vesicle then ruptures, releasing the molecule to the exterior. The vesicles in question are called exocytic vesicles, and the process is called exocytosis or reverse pinocytosis, Fig 1.6. We will now consider some further details about transfer of substances across cell membranes, as endocytic vesicles are derived from cell membrane, and as exocytic vesicles fuse with the latter. There is a constant transfer of membrane material between the surface of the cell and vesicles within the cell. Areas of cell membrane which give origin to endocytic vesicles are marked by the presence of fusogenic proteins that aid the formation of endocytic vesicles. Fusogenic proteins also help in exocytosis by facilitating fusion of membrane surrounding vesicles with the cell membrane. When viewed by M, areas of receptor M-mediated endocytosis are seen as depressed areas called coated pits, Fig 1.7. The membrane lining the floor of the pits is thickened because of the presence of a protein called clathrin. This protein forms a scaffolding around the developing vesicle and facilitates its separation from the cell membrane. Thereafter, the clathrin molecules detach from the surface of the vesicle and return to the cell membrane. The term transcytosis refers to a process where material is transferred right through the thickness of a cell. The process is seen mainly in flat cells, e.g., endothelium. The transport takes place through invaginations of cell membrane called caveoli. A protein caveolin is associated with caveoli, Fig 1.8. Caveoli differ from coated pits in that they are not transformed into vesicles. Caveoli also play a role in transport of extracellular molecules to the cytosol, without formation of vesicles, Fig 1.9. Contacts between adjoining cells. In tissues in which cells are closely packed, the cell membranes of adjoining cells are separated, over most of their extent, by a narrow space, about 20 nanometers. This contact is sufficient to bind cells loosely together, and also allows some degree of movement of individual cells. In some regions the cell membranes of adjoining cells come into more intimate contact. These areas can be classified as follows. Classification of cell contacts. Unspecialized contacts. These are contacts that do not show any specialized features on M examination. At such sites adjoining cell membranes are held together as follows. Some glycoprotein molecules, present in the cell membrane, are called CE/adhesion molecules, CAMs. These molecules occupy the entire thickness of the cell membrane, i.e., they are transmembrane proteins. At its cytosolic end each CAM is in contact with an intermediate protein, or link protein, that appears to hold the CAM in place. Fibrous elements of the cytoskeleton are attached to this intermediate protein, and thus, indirectly, to CAMs. The other end of the CAM juts into the 20 nanometers intercellular space, and comes in contact with a similar molecule from the opposite cell membrane. In this way a path is established through which forces can be transmitted from the cytoskeleton of one cell to another, Fig 1.10. CAMs and intermediate proteins are of various types. Contacts between cells can be classified on the basis of the type of CAMs proteins present. The adhesion of some CAMs is dependent on the presence of calcium ions, while some others are not dependent on them, Fig 1.11. Intermediate proteins are also of various types, catenins, vinculin, actinin. Specialises junctional structures. These junctions can be recognized by M. The basic mode of intercellular contact, in them, is similar to that described above and involves, CAMs, intermediate proteins, and cytoskeletal elements. Junctional areas that can be identified can be summarized as follows. Anchoring junctions or adhesive junctions bind cells together, they can be of the following types. Adhesive spots, also called desmosomes, or max/AE adherens. Adhesive belts or zona adherens. Adhesive strips or fascia adherens. Modified anchoring junctions attach cells to extracellular material. Such junctions are seen as hemidesmosomes, or as Lacaille spots. Occluding junctions, tight junctions. Apart from holding cells together, these junctions form barriers to movement of material through intervals between cells. 1. Communicating junctions, or gap junctions. Such junctions allow direct transport of some substances from cell to cell. The various types of cell contacts mentioned above are considered one by one below. 2. Anchoring junctions. 3. Adhesion spots, desmosomes, maculae adherens. These are the most common type of junctions between adjoining cells. Desmosomes are present where strong anchorage between cells is needed. Example between cells of the epiderm is. As seen by M. A desmosome E is a SM all circumscribed area of attachment, Fig 1.12a. At the site of a desmosome the plasma membrane, of each cell, is thickened because of the presence of a dense layer of proteins on its inner surface, i.e., the surface towards the cytoplasm. The thickened areas of the two sides are separated by a gap of 25 nanometers. The region of the gap is rich in glycoproteins. The thickened areas of the two membranes are held together by fibrils that appear to pass from one membrane to the other across the gap. ////E now know that the fibrils seen in the intercellular space represent CAMs, Fig 1.13. The thickened area, or plaque, seen on T cytosolic aspect of IE cell membrane is produced by the presence of intermediate, link, proteins. Cytoskeletal filaments attached to the thickened area are intermediate filaments. CAMs seen in desmosomes are integrins, desmoglines I2. The link proteins are desmoplakins. Adhesive bellies, zonula adherens. In some situations, most typically near the apices of epithelial cells, we see a kind of junction called the zonula adherens, or adhesive belt, Fig 1.12b. This is similar to a desmosome in being marked by thickenings of the two plasma membranes, to the cytoplasmic aspects of which fibrils are attached. However, the junction differs from a desmosome as follows. A. Instead of being a small circumscribed area of attachment, the junction is in the form of a continuous band passing all around the apical part of the epithelial cell. B. The gap between the thickenings of the plasma membranes of the two cells is not traversed by filaments. The CAMs present are cadherins. In epithelial cells, zona adherens are located immediately deep to occluding junctions, Fig 1.16. Adhesive strips, fascia adherens. These are similar to adhesive belts. They differ from the latter in that the areas of attachment are in the form of short strips, and do not go all round the cell. These are seen in relation to smooth muscle, intercalated discs of cardiac muscle, and in junctions between glial cells and nerves. Hemidesmosomes. These are similar to desmosomes, but the thickening of cell membrane is seen only on one side. As such junctions, the external ends of cams are attached to extracellular structures. Hemidesmosomes are common where basal epidermal cells lie against connective tissue. The cytoskeletal elements attached to intermediate proteins are keratin filaments, as against intermediate filaments in desmosomes. As in desmosomes, the cams are integrins. These are also called /OCA/adhesion plagues, or cocal contacts. They represent areas of local adhesion of a cell to extracellular matrix. Such junctions are of a transient nature, example between a leukocyte and a vessel wall. Such contacts may send signals to the cell and initiate cytoskeletal formation. The cams in focal spots are integrins. The intermediate proteins, that bind integrins to actin filaments, are actinin, vinculin, and talin. Occluding junctions, zonula occludens. Like the zonula adherens, the zonula occludens are seen most typically near the apices of epithelial cells. At such a junction the two plasma membranes are in actual contact, Fig 1.14a. These junctions act as barriers that prevent the movement of molecules into the intercellular spaces. For example, intestinal contents are prevented by them from permeating into the intercellular spaces between the lining cells. Zonulae occludens are, therefore, also called tight junctions. Recent studies have provided a clearer view of the structure of tight junctions, Fig. 1.15. Adjoining cell membranes are united by cams that are arranged in the form of a network that stitches the two membranes together. Other functions attributed to occluding junctions are as follows. A. These junctions separate areas of cell membrane that are specialized for absorption or secretion, and lie on the luminal side of the cell, from the rest of the cell membrane. B. Areas of cell membrane performing such functions bear specialized proteins. Occluding junctions prevent lateral migration of such proteins. C. In cells involved in active transport against a concentration gradient, occluding junctions prevent back diffusion of transported substances. D. Apart from epithelial cells, zonulae occludens are also present between endothelial cells. E. In some situations occlusion of the gaps between the adjoining cells may be incomplete and the junction may allow slow diffusion of molecules across it. These are referred to as leaky tight junctions. Junctional complex. Near the apices of epithelial cells the three types of junctions described above, namely zonula occludens, zonula adherens, and macula adherens are often seen arranged in that order, Fig. 1.16. They collectively form a junctional complex. In some complexes the zonula occludens may be replaced by a leaky tight junction, or a gap junction, see below. Communicating junctions, gap junctions. At these junctions the plasma membranes are not in actual contact, as in a tight junction, but lie very close to each other, the gap being reduced, from the normal 20 nanometers, to 3 nanometers. In transmission electron micrographs this gap is seen to contain bead-like structures, Fig. 1.14b. A minute canaliculus passing through each bead connects the cytoplasm of the two cells, thus allowing the free passage of some substances, sodium, potassium, calcium, metabolites, from one cell to the other, also see below. Gap junctions are, therefore, also called maculae communicantes. They are widely distributed in the body. Changes in pH or in calcium ion concentration can close the channels of gap junctions. By allowing passing of ions they lower transcellular electrical resistance. Gap junctions form electrical synapses between some neurons. A. The number of channels present in a gap junction can vary considerably. Only a few may be present, in which case the junctions would be difficult to identify. At the other extreme, the junction may consist of an array of thousands of channels. Such channels are arranged in hexagonal group B. The wall of each channel is made up of 6 protein elements, called nexons, or connexons. The inner ends of these elements are attached to the cytosolic side of the cell membrane, while the outer ends project into the gap between the two cell membranes, Fig. 1.17. Here they come in contact with, and align perfectly with, similar nexons projecting into the space from the cell membrane of the opposite cell, to complete the channel. Cell organelles. We have seen that, apart from the nucleus, the cytoplasm of a typical cell contains various structures that are referred to as organelles. They include the ribosomes, mitochondria, the Golgi complex, and various types of vesicles, Fig. 1.18. The cytosol also contains a cytoskeleton made up of microtubules, microfilaments, and intermediate filaments. Centrioles are closely connected with microtubules. We shall deal with these entities one by one. Endoplasmic reticulum. The cytoplasm of most cells contains a system of membranes that constitute the endoplasmic reticulum, ER. The membranes form the boundaries of channels that may be arranged in the form of flattened sacs, or cisternae, or of tubules. Because of the presence of the ER, the cytoplasm is divided into two components, one within the channels and one outside them, Fig. 1.19. The cytoplasm within the channels is called the vacuoplasm, and that outside the channels is the hyaloplasm or cytosol. In most places the membranes forming the ER are studded with minute particles of RNA called ribosomes. The presence of these ribosomes gives the membrane a rough appearance. Membranes of this type form what is called the rough, or granular, ER. In contrast, some membranes are devoid of ribosomes and constitute the smooth or agranular ER, Fig. 1.19. Rough ER represents the site at which proteins are synthesized. The attached ribosomes play an important role in this process, page 36. The lumen of rough ER is continuous with the perinuclear space, between the inner and outer nuclear membranes. It is also continuous with the lumen of smooth ER. Smooth ER is responsible for further processing of proteins synthesized in rough ER. It is also responsible for synthesis of lipids, especially that of membrane phospholipids, necessary for membrane formation. Most cells have very little smooth ER. It is a prominent feature of cells processing lipids. Products synthesized by the ER are stored in the channels within the reticulum. Ribosomes, and enzymes, are present on the outer surfaces of the membranes of the reticulum. Ribosomes. We have seen above that ribosomes are present in relation to rough ER. They may also lie free in the cytoplasm. They may be present singly, in which case they are called monosomes, or in groups which are referred to as polyribosomes, or polysomes. Each ribosome consists of proteins and RNA, ribonucleic acid, and is about 15 nanometers in diameter. The ribosome is made up of two subunits, one of which is larger than the other. Ribosomes play an essential role in protein synthesis. Mitochondria. Mitochondria can be seen with the light microscope in specially stained preparations. They are so called because they appear either as granules or as rods. Mitos equals granule, chondrium equals rod. The number of mitochondria varies from cell to cell, being greatest in cells with high metabolic activity, example in secretory cells. Mitochondria vary in size, most of them being 0.5 to 2 pm in length. Mitochondria are large in cells with a high oxidative metabolism. A schematic presentation of some details of the structure of a mitochondrion, as seen by M, is shown in Fig. 1.20. The mitochondrion is bounded by a smooth O-4 membrane within which there is an inner membrane, the two being separated by an intermembranous space. The inner membrane is highly folded on itself, forming incomplete partitions called kerfste. The space bounded by the inner membrane is filled by a granular material called the matrix. This matrix contains numerous enzymes. It also contains some RNA and DNA. these are believed to carry information that enables mitochondria to duplicate themselves during cell division. An interesting fact, discovered recently, is that all mitochondria are derived from those in the fertilized ovum, and are entirely of maternal origin. Mitochondria are of great functional importance. They contain many enzymes including some that play an important part in Krebs cycle, TCA cycle. ATP and GTP are formed in mitochondria from where they pass to other parts of the cell and provide energy for various cellular functions. These facts can be correlated with the observation that within a cell mitochondria tend to concentrate in regions where energy requirements are greatest. The enzymes of the TCA cycle are located in the matrix, while enzymes associated with the respiratory chain and AT production are present on the inner mitochondrial membrane. Enzymes for conversion of ADP to ATP are located in the intermembranous space. Enzymes for lipid synthesis and fatty acid metabolism are located in the outer membrane. Mitochondrial abnormalities. Mitochondrial DNA can be abnormal. This interferes with mitochondrial and cell functions, resulting in disorders referred to as mitochondrial cytopathy syndromes. The features, which differ in intensity from patient to patient, include muscle weakness, degenerative lesions in the brain, and high levels of lactic acid. The condition can be diagnosed by examination of muscle biopsies. The mitochondria show characteristic paracrystalline inclusions. Golgi complex. The Golgi complex, Golgi apparatus, or merely Golgi, was known to microscopists long before the advent of electron microscopy. In light microscopic preparations suitably treated with silver salts, the Golgi complex can be seen as a small structure of irregular shape, usually present near the nucleus, Fig 1.18. When examined with the complex is seen to be made up of membranes similar to those of smooth ER. The membranes form the walls of a number of flattened sacs that are stacked over one another. Towards their margins the sacs are continuous with small rounded vesicles, Fig 1.21. The cisternae of the Golgi complex form an independent system. Their lumen is not in communication with that of ER. Material from ER reaches the Golgi complex through vesicles. From a functional point of view, the Golgi complex is divisible into three regions, Fig 1.22. The region nearest the nucleus is the cis face, or cis Golgi. The opposite face, nearest the cell membrane, is the trans face, also referred to as trans Golgi. The intermediate part, between the cis face and the trans face, is the medial Golgi. Material synthesized in rough ER travels through the ER lumen into smooth ER. Vesicles budding off from smooth ER transport this material to the cis face of the Golgi complex. Some proteins are phosphorylated here. From the cis face all these materials pass into the medial Golgi. Here sugar residues are added to proteins to form protein-carbohydrate complexes. Finally, all material passes to the trans face, which performs the following functions. A. Proteolysis of some proteins converts them from inactive to active forms. B. Like the medial Golgi, the trans face is also concerned in adding sugar residues to proteins. C. In the trans face various substances are sorted out and packed in appropriate vesicles. The latter may be secretory vesicles, lysosomes, or vesicles meant for transport of membrane to the cell surface. The membranes of the Golgi complex contain appropriate enzymes for the functions performed by them. As proteins pass through successive sacs of Golgi, they undergo a process of purification. Membrane-bound vesicles. The cytoplasm of a cell may contain several types of vesicles. The contents of any such vesicle are separated from the rest of the cytoplasm by a membrane which forms the wall of the vesicle. Hesices are formed by budding off from existing areas of membrane. Some vesicles serve to store material. Others transport material into or out of the cell, or from one part of a cell to another. Hesices also allow exchange of membrane between different parts of the cell. Details of the appearances of various types of vesicles will not be considered here. However, the student must be familiar with their terminology given below. Phagosomes. Solid foreign materials, including bacteria, may be engulfed by a cell by the process of phagocytosis. In this process the material is surrounded by a part of the cell membrane. This part of the cell membrane then separates from the rest of the plasma membrane and forms a free-floating vesicle within the cytoplasm. Such membrane-bound vesicles, containing solid ingested material, are called phagosomes. Also see lysosomes. Pinocytotic vesicles. Some fluid may also be taken into the cytoplasm by a process similar to phagocytosis. In the case of fluids the process is called pinocytosis and the vesicles formed are called pinocytotic vesicles. Exocytic vesicles. Just as material from outside the cell can be brought into the cytoplasm by phagocytosis or pinocytosis, materials from different parts of the cell can be transported to the outside by vesicles. Such vesicles are called exocytic vesicles, and the process of discharge of cell products in this way is referred to as exocytosis, or reverse pinocytosis. Secretory granules. The cytoplasm of secretory cells frequently contains what are called secretory granules. These can be seen with the light microscope. With the M, each granule is seen to be a membrane-bound vesicle containing secretion. The appearance, and staining reactions of these secretory granules differ depending on the type of secretion. These vesicles are derived from the Golgi complex. Other storage vesicles. Materials such as lipids, or carbohydrates, may also be stored within the cytoplasm in the form of membrane-bound vesicles. Lysosomes. These vesicles contain enzymes that can destroy unwanted material present within a cell. Such material may have been taken into the cell from outside, example bacteria, or may represent organelles that are no longer of use to the cell. The enzymes present in lysosomes include, amongst others, proteases, lipases, carbohydrases, and acid phosphatase. As many as 40 different lysosomal enzymes have been identified. Lysosomes belong to what has been described as the AC/R/Vesic/E system. The vesicles of this system are covered by membrane which contains HAT posse. This membrane acts as a H pump, creating a highly acid environment within the vesicle, up to pH 5. The stages in the formation of a lysosome are as follows. 1. Acid hydrolase enzyme ES synthesized in ER reach the Golgi complex where they are packed into vesicles, Fig 1.23. The enzymes in these vesicles are inactive because of the lack of an acid medium. These are called primary lysosomes or Golgi hydrolase vesicles. 2. These vesicles fuse with other vesicles derived from cell membrane, infosomes. These endosomes possess the membrane proteins necessary for producing an acid medium. The product formed by fusion of the two vesicles is an endolysosome, or secondary esosome. 3. H ions are pumped into the vesicle to create an acid environment. This activates the enzymes and a mature lysosome is formed. Lysosomes help in digesting the material within phagosomes, described above, as follows. A lysosome, containing appropriate enzymes, fuses with the phagosome so that the enzymes of the former can act on the material within the phagosome. These bodies consisting of fused phagosomes and lysosomes are referred to as phagolysosomes, Fig 1.23. In a similar manner lysosomes may also fuse with pinocytotic vesicles. The structures formed by such fusion often appear to have numerous small vesicles within them and are, therefore, called multivesicular dotties. After the material in phagosomes or pinocytotic vesicles has been digested by lysosomes, some waste material may be left. Some of it is thrown out of the cell by exocytosis. However, some material may remain within the cell in the form of membrane-bound residual bodies. Lysosomal enzymes play an important role in the destruction of bacteria phagocytosed by the cell. Lysosomal enzymes may also be discharged out of the cell and may influence adjoining structures. Lysosomes are present in all cells except mature erythrocytes. They are a prominent feature in neutrophil leukocytes. Genetic defects can lead to absence of specific acid hydrolases that are normally present in lysosomes. As a result, some molecules cannot be degraded, and accumulate in lysosomes. Examples of such disorders are lysosomal glycogen storage disease, in which there is abnormal accumulation of glycogen, and Tay-Sachs in which lipids accumulate in lysosomes and lead to neuronal degeneration. Peroxisomes. These are similar to lysosomes in that they are membrane-bound vesicles containing enzymes. The enzymes in most of them react with other substances to form hydrogen peroxide, which is used to detoxify various substances by oxidizing them. The enzymes are involved in oxidation of very long-chain fatty acids. Hydrogen peroxide resulting from the reactions is toxic to the cell. Other peroxisomes contain the enzyme catalase, which destroys hydrogen peroxide, thus preventing the latter from accumulating in the cell. L-broxisomes are most prominent in cells of the liver and in cells of renal tubules. Defects in enzymes of peroxisomes can result in metabolic disorders associated with storage of abnormal lipids in some cells, brain, adrenal. The cytoskeleton. The cytoplasm is permeated by a number of fibrillar elements that collectively form a supporting network. This network is called the cytoskeleton. Apart from maintaining cellular architecture, the cytoskeleton facilitates cell motility, example by forming cilia, and helps to divide the cytosol into functionally discrete areas. It also facilitates transport of some constituents through the cytosol, and plays a role in anchoring cells to each other. The elements that constitute the cytoskeleton consist of the following: 1. Microfilaments. 2. Microtubules. 3. Intermediate filaments. These are considered below. Microfilaments. These are about 5 nanometers in diameter. They are made up of the protein actin. Individual molecules of actin are globular, G-actin. These join together, polymerase, to form long chains called microfilaments. Actin filaments form a meshwork just subjacent to the cell membrane. This meshwork is called the cell cortex. The filaments forming the meshwork are held together by a protein called filamen. The cell cortex helps to maintain the shape of the cell. The meshwork of the cell cortex is labeled. The filaments can separate, under the influence of actin-severing proteins, and can reform in a different orientation. That is how the shape of a cell is altered. Microvilli contain bundles of actin filaments, and that is how they are maintained. Filaments also extend into other protrusions from the cell surface. Microtubules. Microtubules are about 25 nanometers in diameter, Fig 1.24. The basic constituent of microtubules is the protein tubulin, composed of subunits A and chains of tubulin form protofilaments. The wall of a microtubule is made up of 13 protofilaments that run longitudinally, Fig 1.24. The tubulin protofilaments are stabilized by microtubule associative proteins, MAFs. Microtubules are formed in centrioles, see below, which constitute a microtubule organizing center. The roles played by microtubules are as follows. As part of the cytoskeleton, they provide stability to the cell. They prevent tubules of ER from collapsing. Microtubules facilitate transport within the cell. Some proteins, dynein, kinesin, present in membranes of vesicles, and in organelles, attach these to microtubules, and facilitate movement along the tubules. Such transport is specially important in transport along axons. In dividing cells, microtubules form the mitotic spindle. Cilia are made up of microtubules, held together by other proteins. Intermediate filaments. These are so called as their diameter, 10 nanometers, is intermediate between that of microfilaments, 5 nanometers, and of microtubules, 25 nanometers. The proteins constituting these filaments vary in different types of cells. They include cytokeratin, in epithelial cells, neurofilament protein, in neurons, desmin, in muscle, glial fibrillary acidic protein, in astrocytes. Lamin, in the nuclear lamina of cells. Vimentin, in many types of cells. The role played by intermediate filaments is as follows. Intermediate filaments link cells together. They do so as they are attached to transmembrane proteins at desmosomes. The filaments also facilitate cell attachment to extracellular elements at hemidesmosomes. In the epithelium of the skin, the filaments undergo modification to form keratin. They also form the main constituent of hair and of nails. The neurofilaments of neurons are intermediate filaments. Neurofibrils help to maintain the cylindrical shape of axons. The nuclear lamina consists of intermediate filaments.